Hi, my name is Virginia Zamanka, and this is my microbiome experiment, Tufts University. Today's date is December 11, 2018. Let's get started. The abstract, the gut microbiome is a collection of organisms inside the gut of an animal. It plays an important role in controlling digestion and also benefits the immune system. It is affected in most diseases such as obesity, autism, diabetes, allergies, and so on and so forth. Both of our animals that were tested were flat-coated retrievers, females without any known health issues, and they both consume Mother Humbred biscuits along with table scraps of yogurt and diet brand Vera's Dry. So these are the similarities. They're flat-coated retrievers, had the same exact diet, excessive exercise, they're related, no health issues, same environment. The differences, the age, one is 6 months and one is four and a half. The 6 month one eats grass, and then the 4.5 one does not eat grass. And then this is just like the samples we took. Some to, like the one that's like 6 months, 10 months, whatever. They have they take like coconut supplements, and they play indoor and outdoor, while the 2 year, the other dog takes peanut butter treats and only plays inside. Okay, cool. The introduction, the gut microbiome, as I said earlier, is a collection of organisms and bacteria, and it is present inside the gut of an animal. I basically said this. All this stuff in the introduction is basically the same thing in the abstract, so we're just going to move on. So basically the protocol overview is we are going to isolate the DNA from the fecal samples using the quinum ink and the xyrum ink as well. And then we're going to use the polymerase chain reaction, which we're going to amplify a specific region of the bacteria genomes and try and amplify the DNA while also looking for the nucleotide sequence. So, methods and materials, we're going to use the Quaggan kit to isolate for the isolation of the DNA. So we're going to isolate the DNA from the stool using this kit. And then for this, we're going to use the Xenome kit for the DNA concentration. So we're going to use that to concentrate on the DNA with the Xenome kit column, and then raise the impurities while you elute it with a special buffer, 80% of the buffer. So now we're gonna amplify the DNA. We're gonna amplify certain regions, the 16S ribosome RNA gene, using the polymerase chain reaction, which is also the PCR. And then we're gonna do the gel electrophoresis, which is the technique which relies on the ability of charged particles to solution to migrate in the electrical field. Then we're gonna do the PCR cleanup with the argon core XP kit to clean up the aflatoxin from our PCR reaction. Then, we're going to determine the nucleotide sequence of the variable 16S ribosome RNA. So then, if we do the DNA quantitation, you can come a little closer. So you can tell from our results just the difference from like sample 1 to sample 4. Okay, back up. Okay, so can you come back? I'm sorry. So if you can tell just from this picture how you can tell it was unsuccessful, the PCR cleanups for both samples 1, 2, and 3. Then you can also tell that these are just the results, basically, like the results of the 3 samples. I know only the positive control was the DNA was present, but for the negative control it wasn't. So in the discussion, based on the UV absorbance data, there was not enough DNA for the negative control, and it was not pure enough, but the positive control had DNA, but it was still not as pure. The PCR reactions results that the gel electrophoresis, the samples were unsuccessful. The experimental design and execution were accurate, and our hypothesis was tested and came out to be true. The quantity was not as successful and there was not high concentration. One sample had a super low concentration, on the other sample was 0.343. It did not work in the gel electrophorus, but for the PCR cleanup, we only used positive and negative controls. I forgot to mention earlier that our question was, were two alike animals with different backgrounds have similar gut microbiomes, and our hypothesis was that we're going to identify major changes between the two samples due to the difference in age, food, living, and other animals and indoors versus outdoor. So that's the end of my project. Oh wait, I forgot to say our hypothesis was supported and it was true. Yeah.